Welcome to the deep dive. Today, we're shifting focus a bit. We usually talk about the cell, maybe like a tiny self-contained house, but now we're looking at the neighborhood. How do these houses connect to form tissues, organs, functional systems? So our mission today is to really dig into the source material on cell-cell interactions. We want to explore that intricate microarchitecture that actually makes multicellular life possible, how tissues and organs work seamlessly.
Yeah, thinking about that house analogy again, we're looking at the plumbing, the shared walls, the power grid, the communication lines, basically everything that connects the community and makes it work together.
It really is an amazing level of cooperation when you think about it. We often break biology down to single cells doing their jobs, right? But the way an organ actually functions, like the heart beating in rhythm or the gut acting as this incredibly selective barrier, well, that all comes down to how cells link up, stick together, and talk to each other. And these connections, they're managed by these specialized structures, junctional complexes. And they're immediately relevant, aren't they? I mean, if these junctions fail, the consequences are huge. Like you said, the skin, it's a physical barrier. If those connections weaken, your barrier function is gone. Or the heart, a communication breakdown there means chaos, basically. No coordinated pumping.
Exactly right. So these complexes, they have two absolutely critical jobs. First, they provide the mechanical links, the physical ties, for strength. And second, they facilitate the communication, the receptor-ligand interactions needed for cells to signal each other quickly. And to handle all this, the sources usually group them into three main types. You've got occluding junctions, those are the seals, then anchoring junctions, which are like the heavy-duty bolts holding things together, and finally communicating junctions, which are the direct information channels.
Okay, let's really unpack this. Before we even get to the junctions themselves, we need to talk about the surface they're built on, the plasma membrane. It's not just a simple uniform wrapper, is it? It's actually highly organized.
Oh, absolutely. It's the foundation. And it's incredibly sophisticated. We learned it's a fluid lipid bilayer, sure, but it's far from passive. It's structurally asymmetric, meaning the inside face is different from the outside face, and it's very heterogeneous. The lipids aren't spread out evenly. They form specific zones or microdomains.
Like those famous lipid rafts, right? Why is that unevenness, that organization, so important for making connections?
It's all about location, location, location for the proteins. Lipid rafts are these small, organized patches rich in specific things like glycosphingolipids and cholesterol. The key thing is that different membrane proteins prefer or avoid these raft environments. So the cell uses this to group certain proteins together. This clustering directly impacts which proteins can interact, which affects how a cell connects to its neighbor or to the stuff outside it, the extracellular matrix or ECM. It's like organizing toolkits on the cell surface. If the right tools aren't together, the job doesn't get done.
So the membrane is actively sorting its connection machinery.
Okay.
And then on the very outside, there's that fuzzy layer, the glycocalyx.
Yes, the extracellular coat. It's like a dense forest of complex sugar chains attached to proteins and lipids on the outer surface. It acts as a chemical and mechanical buffer, definitely. But the research highlights something really interesting about its chemistry.
Which is?
Its role in recognition based on electrical charge. The source material points out that certain glycolipids, especially gangliosides, have these complex sugar structures that carry a lot of negative charge. This specific chemical signature, this pattern of charge, is vital for cells recognizing each other.
This sounds pretty abstract, but I guess it has big real-world consequences, like in immunity or reproduction.
Exactly. It's like a cellular ID badge. For instance, during inflammation, immune cells recognize specific charge patterns on the glycocalyx of the cells lining blood vessels. That tells them where to stick and cross over to fight infection. And it's also critical for the very first steps of sperm egg fusion. The specific charge and shape of that coat helps determine if these two cells recognize each other and can start the fusion process.
Okay, so lipid rafts sort the tools, the glycocalyx handles the initial hello. What about the proteins themselves, the actual workers? How they attach to the membrane seems to relate to their job.
That's a really key structural point. The proteins that go all the way through the membrane, the internal transmembrane proteins, they're deeply anchored. These are often the channels, the transporters, the structural pores. They're built for stability. But other proteins are attached more loosely, maybe just on the surface, often with links that can be easily broken. These are much more likely to be involved in quick, temporary signaling events.
So deep anchor for structure, light anchor for quick messages. That makes sense. The attachment reflects the function.
Precisely. Okay, foundation laid. Now let's look at the actual junctional architecture. Hashtag tag 2, mechanical and barrier junctions, the physical links. Right, so when you're building a tissue, especially something like an epithelium that has to separate two different environments, think gut lining separating food from blood, or kidney tubules separating urine from blood, the absolute first requirement is a perfect seal.
Which brings us to the first type. Occluding junctions, or as most people know them, tight junctions. The cellular caulking, maybe?
Caulking is a good way to put it. They are the gatekeepers. Their main job is to seal the space between adjacent epithelial cells. This creates a continuous barrier that's selectively permeable. Critically, this restricts paracellular movement stuff leaking between the cells. By sealing that gap, they force everything that needs to cross the tissue layer to go through the cells themselves, where transport can be tightly controlled.
How does that seal actually work at the molecular level? The diagrams look pretty complex.
It is complex. It forms this mesh-like network, almost like stitches, quilting the membranes of adjacent cells together. The main transmembrane proteins doing the stitching are from the claudin family. Claudins are really interesting because different claudins can create seals with slightly different properties, tuning the junction's permeability. There's also another family involved, the TAMP family, tight junction-associated Marvel proteins.
Okay, so the claudins and TAMP stitch the membranes together, but they must need some kind of internal bracing, right? How do they connect to the inside of the cell?
Oh, absolutely. They need significant intracellular scaffolding. This is where a family of proteins called zonula occludens, ZO1, ZO2, and ZO3 comes in, along with another called cingulin. These aren't just passive anchors, though. They're big adapter proteins that link the transmembrane claudins and TAMPs to the cell's internal actin cytoskeleton specifically, a belt of actin filaments that runs around the cell near the top. And importantly, these ZO proteins also seem to act as signaling hubs themselves, integrating signals from inside the cell.
Wait, so the ZO proteins aren't just like rebar, they're actually involved in signaling too.
It seems so. They sit right at this critical junction, linking the outside seal to the dynamic actin network inside. This allows the cell to actually adjust the tightness of the junction based on internal signals, maybe related to nutrient levels or stress.
That's fascinating. Okay, besides sealing, the source material really emphasizes their role in maintaining polarity. Why is the tight junction the key boundary for this?
Because it acts like a fence within the membrane itself. It prevents membrane proteins that belong on the apical surface, the top surface facing the outside world, or the lumen from drifting down and mixing with proteins on the basolateral surface, the bottom and sides facing the underlying tissue and bloodstream. This separation, this polarity, is absolutely fundamental for specialized jobs like vectorial transport.
Can you give us a quick analogy for vectorial transport? Make it stick for the listener.
Sure. Think of it like a one-way street with security checkpoints. If you're an intestinal cell absorbing nutrients, you need transporters on the apical side to pull glucose in from the gut lumen. Then you need different transporters on the basolateral side to push that glucose out into the bloodstream. Vectorial transport means ensuring the flow is strictly one way. The tight junction acts as the fence, ensuring the indoors apical proteins stay separate from the outdoors basolateral proteins. Without that fence, the glucose could just lead back out the way it came in.
Got it. The junction enforces the direction. Makes perfect sense.
And we have to touch on the dynamic nature of these junctions again. They form this incredible barrier, but they aren't completely static. They have to be able to change. This is crucial for things like wound healing in an epithelium, but maybe the most dramatic example is allowing inflammatory cell migration.
Yes, the immune cell problem. How does a big white blood cell get across this supposedly impenetrable seal?
It has to temporarily breach it. During inflammation, chemical signals tell the tight junction proteins, the claudins, the ZO scaffold, to locally and transiently rearrange. They essentially pull apart just enough to create a tiny temporary gap for the immune cell to squeeze through. The amazing part is that this happens very locally, and the junction seals right back up immediately afterwards, preserving the overall barrier while letting the immune cell get to the site of infection. It's a really controlled, necessary sacrifice. Okay, moving on to the next major category. Anchoring junctions. If tight junctions are the seals, these are the structural anchors, the rivets, the high-strength cables. Their job is pure mechanics, physically attaching cells to each other or attaching cells to the extracellular matrix, the ECM.
And the key players for the cell-to-cell anchoring are the corrosions, right?
Yes. Cadherins are the central transmembrane glycoproteins for cell-to-cell adhesion. They work through homotypic interactions. That means a cadherin molecule on one cell specifically recognizes and binds the same type of cadherin on the neighboring cell. It's like having specific Velcro patches that only stick to identical patches.
And there are two main types of these cell-to-cell anchors based on what they connect to inside the cell. Adherens junctions and desmosomes. Let's start with the adherens junctions. They seem a bit more dynamic.
That's right. Adherens junctions are often found as a continuous belt just below the tight junctions. Inside the cell, the cadherins link up with the actin microfilaments, and because the actin cytoskeleton is very dynamic, constantly rearranging, these junctions are involved in things like sensing mechanical forces, changing cell shape, and cell movement.
So connecting to that dynamic actin network means they're important when cells need to rearrange or move, like during development or wound healing.
Exactly. They provide strength, but also flexibility. And this is where we see a stark clinical connection. The source material really highlights one specific cadherin, E-cadherin, which is crucial in epithelial tissues. It turns out that losing the function of E-cadherin is a key event in allowing some cancers to become invasive. They see this particularly in certain types of stomach cancer and lobular breast cancer. They show this discohesive invasion pattern.
Wow, hold on. So the difference between a contained tumor and one that starts spreading aggressively can hinge on the failure of basically one type of anchoring protein. That seems incredibly critical.
It really underscores how vital these connections are. If you lose that E-cadherin glue, the cells detach from each other, they can change shape, become more mobile, and then they're free to break away from the main tumor and invade surrounding tissues. Understanding E-cadherin failure is fundamental to understanding metastasis.
Okay. Now, what about the other type, the really tough ones, desmosomes? How are they different?
Desmosomes are built for pure tensile strength. They also use cadherin family proteins on the outside, specifically desmogleins and desmocollins. But the crucial difference is inside the cell. Instead of linking to actin, desmosomes connect to the much tougher, rope-like intermediate filaments like keratin in skin cells.
Ah, the intermediate filaments, the cell's steel cables, basically.
Pretty much. This linkage makes the desmosome connection incredibly strong. Its function is to take mechanical stress pulling, shearing forces, and distribute that force across the entire network of cells in the tissue. This is what gives tissues like your skin epidermis or your heart muscle the ability to withstand constant physical stress without tearing apart. Adherens junctions offer adaptable connections. Desmosomes offer brute strength.
That makes sense. Different connections for different mechanical needs.
And lastly, under anchoring junctions, we have the ones that connect the cell not to another cell, but to the underlying scaffold, the ECM. These are hemodesmosomes and focal adhesion complexes.
Hemodesmosomes, literally half desmosomes linking the cell base to the tissue below it.
Exactly. Structurally, they're different because they use a different class of transmembrane connectors called integrins. Integrins bridge the gap between the cell's internal intermediate filaments like keratin and proteins in the ECM, such as laminin. But it gets even more interesting with the focal adhesion complexes that often form around these attachment sites.
Focal adhesion sounds like they're doing more than just sticking. Sensing, maybe.
Precisely. They are incredibly complex signaling hubs. These complexes contain over 100 different proteins all clustered together. Their main job seems to be generating signals inside the cell in response to physical forces or shear stress experienced outside the cell.
So they're like little sensors constantly checking the mechanical environment.
Yes. Think about the cells lining your blood vessels and epithelial cells. They're constantly feeling the drag and pressure of blood flow, or heart muscle cells under strain. The focal adhesion complexes detect that mechanical stress and translate it into biochemical signals inside the cell. This can trigger responses like reinforcing the cytoskeleton or even changing which genes are turned on or off. They're the cell's mechanosensors. #3, communicating junctions, electrical and chemical signals.
Okay, we've got the seals, we've got the structural anchors, but tissues need to coordinate, right? Cells need to talk to each other instantly, much faster than hormones could allow.
That must be where communicating junctions come in. Exactly. Now we're talking about gap junctions. These are the direct pipelines between cells. And unlike the others, they provide essentially zero structural strength. Their purpose is purely communication speed and signaling. They allow the direct passage, the diffusion of small chemical molecules or electrical signals right from the cytoplasm of one cell into the cytoplasm of its neighbor.
How are these channels actually built? What are the molecules?
They're formed by clusters of pores, tiny channels about 1.5 to 2 nanometers across, called connexins. Each connexin is like a little barrel made up of 6 protein subunits called connexins. So one cell puts a connexin in its membrane, the neighboring cell puts a matching connexin in its membrane, and the two connexins dock together end to end, forming a continuous open channel between the two cells.
And that small pore size means only small things can get through.
That's right. It's selective based on size and, to some extent, charge. The source material mentions they allow passage of ions, simple sugars, amino acids, nucleotides, vitamins, small essential molecules, and signaling ions. This allows connected cells to rapidly share metabolites and synchronize their chemical state.
Is it just one type of channel, or is there variety?
Oh, huge variety. That's a key point. The specificity comes from the connexin proteins themselves. Humans have over 20 different connexin genes. The specific mix of connexins that make up the 6 subunits of a connexin determines the exact size of the pore, its charge selectivity, and what it lets through. Different tissues express different connexins, tailoring the communication network. For instance, specific connexins are vital in the lens of the eye, others in nerve cells, others in the heart.
And these channels aren't always open, are they? What controls them?
No, they can open and close very rapidly. They're sensitive to the cell's metabolic state. The source highlights 2 key signals that cause them to close quickly: a drop in intracellular pH, the cell becoming acidic, or a sharp rise in intracellular calcium. These are usually signs that a cell is damaged or dying. So, closing the gap junctions quickly isolates the damaged cell from its healthy neighbors, preventing the spread of toxic ions like calcium, essentially quarantining the problem.
It's a protective mechanism then, cutting off the connection. And you mentioned the heart earlier as a classic example.
Yes, the heart is the textbook case. In cardiac muscle cells, gap junctions provide direct electrical coupling. When one cell generates an electrical signal, an action potential, the resulting flow of ions, particularly calcium, passes instantly through the gap junctions to all its neighbors. This allows the entire mass of heart muscle cells, the myocardium, to contract almost simultaneously, acting as a single functional unit, a syncytium. Without that rapid communication via gap junctions, the heart couldn't beat effectively. It would just quiver uselessly, which is fibrillation. #4, the cytoskeletal scaffold. Before we move on completely from the architecture, we really need to circle back and emphasize how all these junctions, the seals, the anchors, the communication ports, are totally dependent on the cell's internal framework, the cytoskeleton.
Right, the internal scaffolding. It maintains the cell's shape, organizes everything inside, allows movement. The junctions are really just the points where this internal system connects to the outside world or to other cells.
Exactly. And which cytoskeletal element a junction connects to tells you a lot about its function. Let's start with the heavy-duty ones, the intermediate filaments.
Those are the strong ropes we talked about linked to desmosomes and hemidesmosomes.
Correct. They form these very tough, rope-like polymers. Keratins in epithelial cells are a prime example, or vimentin in cells from connective tissue. They provide the serious tensile strength needed for desmosomes to anchor effectively and dissipate mechanical forces across the tissue. When your skin resists friction, it's largely thanks to this network of keratin filaments connected by desmosomes absorbing the stress. It's a brilliant design. Connect a stress-resistant junction to a stress-resistant internal cable network.
And if that system fails, if those connections break under stress,
You get major problems. Genetic defects in keratin proteins, or in the integrins that link keratin to the ECM via hematosomes, cause severe blistering diseases. Even normal friction can cause the skin layers to shear apart because the mechanical linkage is compromised. It really shows how vital that tensile strength is. Interestingly, the source also notes that the specific type of intermediate filament protein expressed by a cell is tissue-specific. Pathologists actually use this to help identify the origin of poorly differentiated tumors, like finding vimentin suggests a mesenchymal origin, finding cytokeratins points to an epithelial origin.
so they're structurally critical and diagnostically useful. Okay, now contrast that with the other main player, the actin microfilaments, right? The thinner, more dynamic cables associated with adherens junctions.
Actin is all about dynamics and change. These filaments are thinner and constantly being built and taken apart, a process called actin treadmilling, which uses energy. This constant remodeling is what allows cells to change shape, to crawl or migrate, and it also allows the cell to actively control the tension and position of the adherens junctions themselves. Think of actin as the adjustable, repositionable scaffolding, while intermediate filaments are the permanent load-bearing beams.
It really seems the cytoskeleton is the integrator, pulling together mechanical forces, structural support, and movement, all mediated through these highly specific junction points. #HAG implications and future perspectives. Okay, let's shift from the how it's built to the what happens when it breaks. The link between these connections and disease seems incredibly direct. What does studying this cellular connectivity really tell us about major illnesses?
Well, it's becoming increasingly clear that failures in cell connection are often really early critical steps in major disease processes, especially cancer. We talked on E-cadherin already, but it bears repeating. The loss of this single anchoring protein in epithelial cells is a classic trigger for tumors becoming invasive. When cells lose E-cadherin, they undergo a transformation called EMT, epithelial mesenchymal transition. They basically shed their stable epithelial identity, become more migratory like mesenchymal cells, and can then break free and invade. The breakdown of the architecture literally enables the spread.
So figuring out how to maintain or restore those connections could be a major therapeutic strategy in cancer.
Absolutely. It's a huge area of research. And studying these kinds of defects effectively really relies on having the right tools. The source material specifically points out how CRISPR-Cas9 gene editing has revolutionized this field.
Right, CRISPR. Can you elaborate a bit on how that specific technology helps us move from just observing these connection failures to actually modeling them and figuring out the mechanisms?
The amazing thing about CRISPR-Cas9 is its precision. It uses a guide RNA molecule to direct an enzyme, the Cas9 nuclease, think molecular scissors, to a very specific spot in the cell's DNA. It then makes a clean cut. The system is incredibly specific, much more so than older methods.
And how does that precision help with studying something as complex as cell junctions?
It allows research to go way beyond just deleting a gene entirely. You can use CRISPR to make very precise edits. For instance, you can introduce a specific point mutation, a single letter change in the DNA sequence that's known to cause a particular disease in humans. So, say there's a hereditary heart condition linked to a tiny change in a connexin gene affecting gap junctions. Researchers can use CRISPR to engineer that exact same mutation into cells in a lab dish or even into a mouse model. Then they can study exactly how that specific molecular change messes up the gap junction's function. Does it change permeability, electrical coupling, response to pH?
So you can basically build the precise disease mechanism into a model system to understand why it causes problems.
Exactly. You could do the same for integrins to study how cells fail to sense mechanical forces in vascular disease, or introduce specific mutations into E-cadherin to watch the very first steps of cancer invasion unfold in a controlled way. This ability to precisely model interaction defects is massively accelerating our understanding of these complex diseases. #tag conclusion.
Wow. Okay, so this deep dive has really been a journey through the intricate architecture of our tissues. We started with the cell surface itself, the plasma membrane, not uniform but organized with things like lipid rafts. Then we moved through the three main types of junctions, the tight junctions forming the essential seals, the anchoring junctions providing mechanical strength with adherens junctions linked to dynamic actin and desmosomes to tough intermediate filaments, and finally, the gap junctions acting as direct communication channels.
And we saw how all of this external architecture is intimately linked to and dependent on the internal cytoskeleton, the strong intermediate filament ropes, and the dynamic actin cables. I think the main takeaway is the incredible specialization and dynamism here. Multicellular life, the function of our organs, it all hinges on this complex social network between cells being perfectly maintained, constantly adapting to both physical forces and chemical signals.
It really is biological engineering at its most sophisticated level.
It truly is. And maybe as a final thought for you, the listener, to mull over. We talked a lot about tight junctions creating this almost impermeable barrier, forcing things to go through cells. But let's revisit that immune cell problem one last time. Think about an infection, say, in your gut lining. White blood cells circulating in your blood need to get across that sealed epithelial layer to fight the bacteria. They perform what's called transepithelial migration. So the question to ponder is this. Considering how complex tight junctions are claudins, TAMPs, zeoproteins, the actin links, what specific signals does the immune cell itself likely use to locally and temporarily persuade that junction to open up just for it? And how does the epithelial cell immediately know to reseal that breach once the immune cell is through to prevent widespread leakage? That constant negotiation between maintaining an absolute barrier and allowing essential access for defense, and that's really where so much of our health plays out.